The only time I'm called Robert is when I'm in trouble with my mother. So I'm Bob Hutchens, President and CEO of StemBioSys. First of all, I want to thank the conference organizers for including me here today, and I'd like to thank all of you for taking time out of a very busy schedule to spend a few minutes listening to our story about StemBioSys. Okay, good. Want to start off with just a quick introduction. The technology that we're going to talk about today we call high-performance microenvironment. This is a decellularized extracellular matrix. I'll go more into that in a second. The technology came out of the University of Texas Health Science Center in San Antonio. We had 3 pending patents when we took the technology out. Today we have 9 issued patents. We have about a dozen pending patents in addition to those 9 issued patents around the world today. From an investor's standpoint, and this is an investor conference, so I want to touch on a few of those points. The core technology, the HPME technology, is really designed to address the need for large numbers of high-quality stem cells. Secondly, if we think about that technology and what we might do with that, we have developed a business plan that is designed to drive very near-term revenue and, in fact, bring that piece of the business to a break-even cash flow in some kind of reasonable time frame. Third, the business plan itself is hedged. This is a platform technology, and there are a number of applications that could benefit from it. And therefore, what we who have in the company and as I've gone out and talked to investors, one of the things we've emphasized is that this is not a binary outcome investment that people are making. There are a number of different applications that may work. If one doesn't, there may be another place that it does, which is when I looked at this company first as an investor, that was one of the things that was particularly attractive to me. The platform itself, the fourth item up there, may have some interesting therapeutic applications in and of itself, and obviously from a long-term value creation, that could be very, very interesting, and I'll talk more about that as we get to the end of this. And then finally, the company itself has a very strong board of directors and management team that we have assembled. So let me just give you some background. I've got a couple slides on the technology itself. I'll talk about where we are taking that technology, and then I'll spend the end of this presentation really talking about the strategy we've been following in the company over the last 2 to 3 years that I've been part of it. So I want to start with the evolution of cell culture, and I think it's important to remind ourselves how this began. We started in the 1940s culturing cells in jars. Into the 50s, we moved to glass petri dishes, and as we got into the 60s and 70s, we began using polystyrene. Today, we submit that the way to our product, the high-performance microenvironment, is the new best way and the next natural evolution in the culture of stem cells. The easiest way to understand our product is for us to share with you our secret and how we make it. And we start with commercially available bone marrow mesenchymal stem cells. We seed those cells into a dish on the top there. We add media and growth factors. Those cells expand. We remove those bone marrow mesenchymal stem cells when they have completed the matrix, and what we have left is a decellularized extracellular matrix at the end of this. If you're interested in that process, there is a video on our website that explains that in a little bit more detail, but I think you get the idea. The reason we're so excited about this, and this was the initial data that we developed, when we compare the technology to other cell culture technologies, we see more cells. But more cells in and of themselves is not interesting if those cells aren't good. And in fact, the cells that come off this matrix are smaller in size. And the gene markers, it's got some really unique gene markers. One that we feel is very important is stage-specific embryonic antigen 4, and we see consistently higher levels of cells that are expanded on our matrix, consistently higher levels of SSEA4 than we do on other technologies. So that was, when I came in a year ago and talked about the company, that was really what we had to talk about at the time. As we've looked forward and over the last year, we have really focused on starting with that core technology. And if you look at the bottom, we talk about the HPME in the research market. So we've sold it, have begun to sell that into the research market, but really have been focused on taking that core product and expanding that and using that as a basis for a number of new products. And the first thing I'll talk about are follow-on HPME products. So other cell matrix products. And most importantly, in the fourth quarter of this year, so sometime in the next 8 weeks or so, we will be announcing a xeno-free version of this technology. Current product, we use fetal bovine serum to make it, but the version we'll be announcing in late November, December will be completely xeno-free. In the first quarter of next year, we will have what we call a free protein version, or it's a version of that xeno-free matrix in a vial. And that has a number of applications if you think about combining that with other products, combining that with using it in a bioreactor, a number of really interesting technologies and applications for that. And then finally, there are 2 cell lines that we have identified and have been working on over the last year. I'll talk about the second one first, and we'll come back to the other one in just a second. In January, we'll be announcing a perinatal-derived stem cell with some very interesting characteristics. Most importantly, we have some early evidence that this cell may in fact be pluripotent. And so stay tuned. We're very excited about that. And as I say, I suspect we'll have an announcement around the time of JP Morgan on that one. The other cell line that we're announcing today is a human cord blood-derived progenitor endothelial cell. And this is a really interesting cell for a number of reasons. If you think about it, we describe it that it sits in between a pure stem cell and a HUVAC, and so it really is a progenitor cell. Similar to HUVAC, but some important differences. At least 20 population doublings these cells are capable of without any diminishing of potency. We also see an ability to form secondary and tertiary colonies, continuing to see colony formation after many, many passages of this cell, versus HUVACs, in which we see colony formation only in the primary formation. And the cell itself is very highly angiogenic. I mean, we see the angiogenesis in a matter of hours with this cell when we have used it. So very interesting, very excited about this cell, and I will make an offer that if any of you are interested, we would be happy to provide you a sample to try this out. So one of the questions I get from investors is, so how big is this tool market that we're talking about? And we've just taken a stab at it here. This is from the 2016 Bioinformat study. The market itself globally is about $4.5 billion. We play in a couple areas of it. If those couple areas represent about 5% of the market, we're going after a $230 million market. The point being here, we don't need a whole lot of that market to have a nice, viable business on our hands. So we're pretty pleased about that. From a strategic standpoint, I really want to shift to sort of where are we taking all of this. There's really 3 elements of the strategy of the company. First and foremost is to commercialize the HPME itself, and that includes all of the products I've just talked about. Initially into the research market, but we certainly see clinical applications longer term. To do that, we've put together a global network of distributors. Again, that was something I couldn't talk about a year ago. We now have that in place today, and I'll show you more on that in just a second. And we are very open and aggressively pursuing inorganic options to bolt on other regenerative medicine tools into our portfolio that we can take to the market and leverage the infrastructure we've put in place. Second, because this is a platform technology, having key collaborations and leveraging those key collaborations going forward. Our 2 cell lines came out of collaborations. That'll continue to be an area of focus. It's why we're at meetings like this, and it's why we talk to partners all the time. Where it makes sense, we will make those collaborations closer, we'll work closer with people, et cetera. And then finally, on a therapeutic standpoint, we will continue to assess the therapeutic benefit of it. Our current strategy is to rely on grant funding to do that, to get it to a point where we've got a financeable therapeutic solution that we can take to the market and raise money around that particular solution. I'll talk more about that in a second. So just going to take these in turn. So the first important thing is from a distribution standpoint, I like to joke, we spent a nanosecond debating about building our own sales force and concluded that was not going to work. And so what we have done is selectively partner with a group of distributors around the world. Our major distributor is VWR here in the United States. We have an exclusive agreement with them for the US and Canada. We anticipate that will get rolled out across the world. We have agreements in Europe We have an agreement with Funakoshi Company Limited in Japan, and we have an agreement with Solin Bioscience in South Korea. So I think we've got in place a good footprint, and we've got a plan to take that to most major markets around the world in the next couple years. From a collaboration standpoint, just to give you a flavor of what our collaborations have been like, we've kind of highlight these. So HPME is at the core of all of these. And the first area we've collaborated with people is anybody who's got a hard-to-grow cell line. And we've worked with people on cancer cells, a whole range of things that we've worked with people on. Our HPME may be a potential solution. And again, this is a situation we're happy to provide these as samples to people to try out, get some initial data, and then we can go from there. We've also actually worked with a couple of companies around diagnostic applications. This tends to be where we've taken our matrix and we've combined it in some manner with an existing product and improved the performance of that product. And then finally, we have a collaboration, one I can talk about in San Antonio with a company who had a polymer stent, and we grew the matrix on the stent and improved the performance of that stent in endothelial growth pretty substantially. So that's an example of combining the technology with a medical device. And then from a therapeutic standpoint, we've done one rigorous study where we took the product, we removed it from the dish, so we had free protein, if you will. We combined that with hydroxyapatite, and we looked at bone growth. And if you look at the 8-week bar there, the combined product is all the way at the end. You can see that there was some reason to believe that the combined product might in fact have superior performance. Our plan at this point is to do a more rigorous study. We've identified some things that we would change in terms of concentration levels, et cetera. And what we are doing now is we've got several government grants in around this to take this to the next level. I said the team, and investors tell me all the time, one of the things I'm investing in is the team. We have a terrific board of directors, headed up by our chairman, Kathy Burzig. Kathy was formerly the CEO of Applied Biosystems in the Bay Area, and then KCI in San Antonio, rounded out by a number of other business people, 2 of our venture capital partners. And from a scientific standpoint, we're very fortunate to have Robert Langer, who I think we can all agree is one of the leading biomaterialists on our board as well. From a financing standpoint, we've raised about $13 million for the company since its inception 4 or 5 years ago. Most recently, we launched a $3 million Series B round of financing. I wish I could tell you we were done. We are literally at $2.9 million. So if anybody wants to write a check for $100,000, we'll be happy to take it and take care of that. But we're very pleased. The company has about 2+ years of runway in place. We run it we're very efficient about how we run it, and we do have a cost competitive advantage due to our location in San Antonio, both from a labor standpoint and from a facility standpoint. To wrap it up, we've got an innovative technology in regenerative medicine, a number of outstanding partners that we have worked with that have led to a very robust pipeline. We're very excited about what we see coming in 2018 and beyond. And we've got some interesting upside in the company as you think about the therapeutic benefits. And that was it. Thank you. We will mention we have a booth at the session tomorrow. If you'd like to learn more, please feel free and stop by, and my colleagues will be happy to share some more information with you. Thank you.